I want to acknowledge my co-authors, Jamie Parham and Larry Keene, and I'll talk a little more about Jamie's contribution as we get into it. I want to just start out by looking at the process of where we've been in genomics and how we got to where we are. We've been working in the area of genomics and beef cattle for a long time, but early on it was not at all clear that we were going to report DNA test results as something that was incorporated into EPDs. There were a lot of different systems that were used in the commercial DNA tests, numbers of stars and 1 to 9 scales and various things like that. And it was obvious to some of us early on that the best way to do it was to wrap them up in the EPDs, but it wasn't at all obvious how and when we were going to get there. And it's really nice that that is where we are now. There have been a lot of challenges along the way. I want to point out that there's at least 9 breeds that currently are reporting genomically enhanced EPDs. I think that's a tremendous step forward from where we were 5 years ago, and when maybe we had 1 breed, and a huge difference from 6 or 7 years ago when we had no breeds doing it. So we've come a long way in what in retrospect seemed like a fairly short time. But as I mentioned, there have been some challenges in incorporating genomic information into EPDs, and some of the challenges that we've faced, particularly in beef cattle, have been getting the scaling of the genomic information right. So the genomic part of our prediction doesn't always come out cleanly in the same units as our traits measured in. So if we're talking about yearling weight, for example, the genomic part may look like it's accounting for a bigger effect than it really is. And that's the thing that we typically would see, is that the genomic effect looks bigger than it really is. And then we have the question of if we're blending genomic and non-genomic information together, and by non-genomic I just mean pedigree and our traditional phenotypes, then how much emphasis do we put on those 2 parts? And in some analyses or methods of developing genomically enhanced EPDs, it's really obvious where this weighting comes in. And in other types of approaches, it's not so obvious, but the weighting is still going on behind the scenes. And when we have these issues, they're both generally attributed to a phenomenon known as discovery bias, which is kind of the main topic of my presentation. I'm going to come back and explain what that is in a few slides. But before I do, there's another challenge that we could deal with, and that's what I'd call genotyping bias, which is something that could occur where just the fact of genotyping an animal might make it look better on average than if it had not been genotyped. And that's probably a consequence of selective genotyping. It's from picking out the animals that we think are the best in our program, spending the money to genotype them, but not genotyping the whole population. If we genotype the whole population, then on average the genotyped ones are probably going to be the same as the non-genotyped ones. But that's not where we are currently in genomic prediction in beef cattle. So this is something that we've had to worry about. And there's been excellent work done to try to address all 3 of these issues that I've described so far. And they've all caused a lot of headaches along the way. They're causing a lot less headaches now than they used to. I think there's still a lot of room for improvement in how we deal with all of these issues. And so I want to just throw out some things that we're thinking about to try to do some of those, to make some of those improvements. But before I do, I want to mention that there's a lot of different ways in which genomic enhanced EPDs have been implemented in beef cattle, and in which genomic evaluation or genomic prediction has been implemented across livestock species in general. I think what's most fundamentally different to me is an assumption of the underlying model that we use for genomic prediction. And the issue is it a weighted analysis or an unweighted analysis? And by a weighted analysis, I mean that the markers, the genetic markers that are thought to be more influential on the trait of interest are given greater emphasis. And so we're weighting the markers based on how important we think they are. And then the alternative to that is to have an unweighted analysis in which we basically assume that all markers are given equal emphasis. And there's a lot of implications as to which of those two models we choose. And I'm going to spend most of the rest of the talk describing what some of those implications are and what we might do to take advantage of some of the advantages and mitigate some of the disadvantages. Before I do that, I want to talk about what do we know about the biology of how different markers influence traits of interest. And this could be any trait that we want to think about selecting, or almost any trait. Some of them would be influenced by a lot fewer genes. But in general, we're going to have a few genes with large effect and many more genes with progressively smaller effects, down to genes that have very small effects, too small to measure. And actually a lot of the genetic variation may be accounted for by genes whose effects individually are too small for us to discern and measure. And you know, we could go from one trait to another and the distribution of these effect sizes will change, and we could go from one species or population to another and they'll change. But the general pattern here, I think, is not really in question among scientists, that we have few genes with large effect and a lot of genes with small effect. And that's just a fundamental challenge that we have to deal with in terms of genomic prediction. So I mentioned discovery bias earlier. Discovery bias is a phenomenon in which the genomic predictions appear more accurate than they really are. It occurs when we use the same data to do two different things. The first of those is to determine which markers are thought to be more influential, and the second one is to actually predict genetic merit conditional on which markers we thought were more influential in the first step. And so discovery bias is the price that we pay for using the same data to do those two different things. It's most problematic when predictions are based on marker effects estimated from data on close relatives. And it's a phenomenon that we think occurs when we do a weighted analysis. We generally assume that it probably doesn't occur when we do an unweighted analysis. So in unweighted prediction, the part about that that's really troubling to me is it's just based on this completely unrealistic assumption that all parts of the genome contribute equally to every trait. And we just know biologically that that's not true. And that assumption has some consequences, like that we would expect the genetic correlations among all traits to be identical, to be 1. And we know that doesn't happen. And so I think we can do better than an unweighted analysis, but unweighted analyses are used very extensively throughout genomic prediction in livestock. And most specifically, that's what essentially what all the dairy evaluations are based on. It's what what a lot of the large swine and poultry breeding companies use in their genomic evaluations. And so they do work. They can be made to work at least. But they work best if the animal to be predicted is closely related to animals, to close relatives that have high accuracy genetic evaluations. There's enough phenotypes close to the animal to be predicted, especially if there's enough progeny and grandprogeny of those, or if the parents and grandparents of those animals to be predicted have a high accuracy genetic evaluation. Under this model, the accuracy is going to decrease pretty rapidly as the relationship of the animal to be predicted to the discovery population decreases. And that's a challenge for us in beef cattle that doesn't exist in the dairy industry in particular, and in these large breeding companies. If they can genotype their whole populations, they get around that problem that way. So unweighted prediction is essentially based on estimating the genomic relationships of the animal to be predicted to many other animals that have high accuracy predictions. And if those relationships to other phenotyped animals are all low relationships, then it's pretty hard for that relationship matrix to be informative. So in beef cattle, we've tended to use weighted predictions. And I would argue that they're generally more accurate than unweighted, but the price for weighting is discovery bias. And so that's the downside to using a weighted analysis. And this conclusion that they're generally more accurate is conditional that the analysis accounts appropriately for all the sources of information. I don't know that we've ever done an analysis that really meets that criteria, but I think we should aim to get closer to that as we move forward. But if we're going to do a weighted analysis, then we need to account for discovery bias. And I want to make an aside here and discuss how we would evaluate the accuracy of genomic prediction. There's fundamentally two different categories of ways, contexts in which we could look at the accuracy of genomic predictions. Early on, we looked at it primarily as a population statistic. So we said, how accurate is this genomic test? And it was a property of the test, not a property of an individual animal. And as we've moved forward, we're beginning to ask the question, well, how accurate is the genomic prediction of a specific animal within a population? And those are related questions, but they're quite different in terms of how we would calculate those and what they mean. But discovery bias can inflate those nominal statistics for either kind of accuracy. When we calculate the accuracy of genomic prediction of an individual animal, we do it in much the same way that we'd calculate the accuracy in a non-genomic genetic evaluation through looking at the inverse of the mixed model equations. But to do that in a genomic context requires then that the discovery data is analyzed simultaneously with the prediction, or else we do some kind of computational trick that basically is trying to mimic doing an analysis that includes both of those. In the old days, we never knew what the discovery population was. We never knew who the animals were in the discovery population. It wasn't possible to do this. But as the technology and the way in which it's being implemented has evolved, we're now often in a situation where we can know the pedigree of the discovery population, what the identity is, have the information to analyze simultaneously. And so this has been a pretty good step forward in terms of looking at accuracy of genomic predictions. But in the old days, we thought about our problem as being composed of three different populations, a training or discovery population, and then another population that we'd validate in, and then yet another population I've listed here is the application population. Those are the animals that we actually want to predict so that we can select in them. And the idea was that we wanted for the genetic correlation or the average relationship between the training population and the validation population to mimic as closely as possible the relationship between training and our application population. And so a tool that was developed and has been used very extensively at Iowa State to try to achieve that equivalence is a technique called k-means validation. And so this would be an example with k equals 3. So we break the population up into 3 different groups, generally trying to make the animals within each of those groups as similar as possible, but not necessarily always grouping them by genetic similarity. And we take all but one of these groups and consider them to be the discovery population, and then estimate the effects of all the markers in the discovery population and apply those marker effects to the validation population and ask, well, now how well did our predictions actually correspond to the phenotypes within the validation population? And this is the approach that basically has been used to provide a lot of those corrections to the various methods of calculating genomically enhanced EPDs that I discussed earlier, in terms of getting the scaling correct and getting the relative weighting on genomics versus non-genomics correct. And so in k-means validation, we would start out, for example, with this population in the green oval as being the validation, but then we make every other one of the groups the validation population, and then when we pool those measures of how accurate the test is across each of the different groups. And so this has been a very effective strategy for helping to correct for discovery bias. And so that's really important. One of the things I want to mention here is, though, that requires doing a separate analysis for each of the different groups that we've got, in this case 3, so we're doing the analysis 3 times. Now the method is more powerful as the number of groups becomes larger, because you don't need a real large number of animals in the validation group, but you need a lot of animals in the discovery subset. So we could make the validation population smaller and smaller and gain efficiencies to some extent. But that is, as long as we're doing a separate analysis for each group, that's going to increase the computational effort on the discovery side. With 3 groups, it's not enough additional computation to really worry about. So how does all this apply to you? There's a lot of different methods that are being used for genomic selection across various breed associations, and they provide these genomically enhanced EPDs that I hope a lot of you have come to rely on. I think they're a really great tool that we've been waiting for a long time, and that now have really come to fruition. I want to discuss now Jamie Parham's thesis project. She did a great job on a really challenging project that was aimed at trying to account for discovery bias in genomic selection in order to improve genomic prediction. And Jamie did her project at South Dakota State and spent about a year here in 2 different times working with Larry Keene and me on the project, and we're fortunate now that she is doing her PhD at University of Lincoln, and so we've got her still close by, even though we're not working with her on a daily basis at this point. But so in Jamie's project, we decided to try taking k-means validation to the limit. And the limit is that each animal is its own group. And And so we use all but one to do the discovery, and then we do the validation and prediction both on each animal. We estimate marker effects conditional on the big subset of the population, and then we apply those marker effects to the animal that we excluded from the analysis of the marker data. And we just rotate through the population, through the animals that are in the population, and conceptually do that for each animal in the population. Now, I mentioned earlier that that could be pretty challenging from a computational standpoint, but we worked out a way that if we do the analysis for the entire data set, then dropping each animal or small group of animals out of that full analysis is pretty computationally trivial. And so working through the whole population in that way is still very feasible from a computational perspective. And that's what I just said there. So what we learned in Jamie's project is that when we went through all this process, the nominal accuracy values that we computed with this method, after accounting for discovery bias, were quite a bit closer to the true accuracy values than those that were computed without accounting for discovery bias. And I thought I had a slide in here, but I must have missed it. Jamie's project was basically based on a simulation study so that we knew what the true breeding values were, we knew what the true accuracies were, so that we could evaluate that. So the accuracy values improved quite a bit after accounting for discovery bias in this way. But more importantly, the true accuracies of the predictions improved a little bit. They didn't improve as much as we'd hoped they would, but they improved some. And in the end, that's really what's important, is we want more accurate predictions. It's nice to know how accurate they are, but the most important is that they're more accurate. So we went through a lot of contortions to get to this point, but we're pretty pleased with what we saw. We still have a lot to learn about it, but we think it's an approach that's worth taking further. So to close, my conclusions are that done properly, and the done properly part is really important here, but done properly, weighted genomic prediction will outperform unweighted in terms of accuracy of the final predictor. To whatever extent this has not yet been achieved, it suggests to me that it'd be fruitful to improve our models and methods of prediction, because I think when we've really taken advantage of the opportunities that are out there, I think this is going to be true in the end. Now I'm going to go talk about this in Fargo on Thursday with a group of statistical geneticists, and there's going to be at least one and probably quite a few in the group that are going to take issue with this conclusion. But my retort to them is, you know, we haven't still fully taken advantage of the opportunities that are there with the weighted analysis. And then I think that we are finding that these methods can be computationally feasible, and that's important, because if we can't compute it, then it's not relevant. But that doesn't seem to be a problem for us. You know, this advantage to weighted prediction is going to be a lot more important for traits where phenotypes are limited. And I don't think we're seeing any real interest from the dairy industry in moving towards these, and don't really know about the big swine and poultry companies, but I'm not aware of interest from them in moving in this direction. But I think that if the dairy industry wants to evaluate feed efficiency and they want to measure phenotypes on a few thousand cows and have accurate genomic predictions, they may need to look at what the beef industry's been doing, because I don't think their approach is going to work very well for that. So as a final thought, I'm going to throw out that I think beef breeders should be proud that we're using a more sophisticated analysis than our counterparts in the dairy, swine, and poultry industries are. You know, theirs is easy to write a computer program for and turn the crank and forget about it. And ours takes more tweaking, and we've got more development still to do. But I think given the limited number of genotypes that we've had to work with, and that we necessarily have to go through in order to get to large numbers of genotypes, I think the path that we've taken in beef has been the correct one. And I'll just mention, you know, the Angus Association is at a point where this number of genotypes is starting to become a lot less of a problem, and probably not contributing so much to discovery bias. But every breed has had to go through a stage where genotypes are limiting. And some of the breeds can't even take the path that was taken by the Angus breed, because what really got the Angus breed there was having genotypes on a large number of high accuracy sires. And the ironic thing for our smaller breeds is they can't take that path because there just aren't enough high accuracy sires in the breed to do it. So the only path for them to take is genotyping lots of low accuracy animals. And so to get there, you know, we've got to use methods that are more powerful than the unweighted analysis. So I'll close there and try to address any questions that you might have.